Okay, let's talk about the complement system. The first thing to know about the complement system is that it is generally considered to be a part of the innate immune system, though it does have some crosstalk with the acquired immune system. The second thing to know is that it is not particularly controlled by cellular agents. It is a blood protein system. So your blood consists of what are called formed elements, which are the cells and cell derivatives like platelets, white blood cells, red blood cells, and the plasma, which contains water and lots of proteins and nutrients and all of the other stuff that is in cells. The complement system is a natural part of the plasma. It's this group of blood proteins that are always present but only become activated under a certain set of circumstances. And these proteins have antimicrobial capabilities. So, first thing to know is that there are three different ways that the complement system can become active. The first is what's called the classical pathway. And this was the first pathway that was discovered, and it is basically controlled by antibodies. And this is where the crosstalk with the acquired immune system comes in. One of the ways that antibodies can act to protect you is to activate the complement system against bacteria, usually bacteria, that it labels. So that's a thing that antibodies can do. They activate the complement system through the classical pathway. The second pathway that can activate the complement system is called the lectin pathway. Lectin is a word that means a protein that specifically binds to sugars. And in this case, the lectin involved binds to mannose, which is a type of sugar that is frequently found on the outside of bacterial cells, but generally not on the outside of your cells. So this mannose binding lectin is always floating around in your blood. And when it binds to mannose on the outside of bacterial cells, it can activate the complement system and direct the complement system against the cell that it has bound to. The third pathway is called the alternative pathway, and this is the main pathway. I know, it's a little weird that the alternative pathway is the main pathway. But the thing is, it was the second pathway that was discovered. The classic pathway was the first one, and people assumed that that was the main pathway that was activated because they found it first. And they found the alternate pathway, and they called it the alternate pathway because they assumed that it was an alternate. Then later on, we discovered that the alternate pathway is actually the main pathway. And it's confusing, but you gotta know it. The alternate pathway works through a positive feedback loop. So all of the complement proteins are labeled like C something, C1, C2, C3, that sort of thing. And a main protein in the complement system is C3. C3 will naturally fall apart and split, but only a little bit. A very small amount of C3 is always just sort of like splitting into two halves called C3A and C3B. I'm going to get into the specifics of how these proteins work later on. But what you need to know for this is that C3b can form something called C3 convertase. And C3 convertase stimulates more C3 to break into C3a and C3b. C3b can only turn into C3 convertase if it embeds in the membrane of a bacterial cell. So you always have a little bit of C3b just naturally floating around in your blood because C3 will sometimes just split randomly. But in the presence of bacteria, C3b embeds in the bacterial membrane and forms C3 convertase, which causes more C3 to split into A and B. That additional B also binds to bacterial membranes, forming more convertase, which makes more C3b, which binds to more bacteria, making more convertase, causing more C3b. And what you have is a positive feedback loop that very quickly spirals to a high number. Once you get, and this is basically the activation of the alternate pathway, because the whole point of the alternate pathway, the whole point of the complement system, as we'll see, is to get more C3b. So we have three ways that the complement system can become activated: by antibodies, by lectins, and by positive feedback triggered by the binding of C3b to foreign cells. By the way, why doesn't C3b bind to your cells? Well, sometimes it does, but your cells have basically a protein in their membranes that tosses C3b back out when it gets embedded in. So no matter which pathway you take to activate, all of these pathways are going to converge on the formation of C3 convertase, whether it's triggered by lectins, by antibodies, or by the positive feedback loop. No matter what, you get C3 convertase formed. And C3 convertase turns C3 into C3a and C3b. There are three ways that the complement system acts to protect your body. These three ways have nothing to do with the activation pathway. No matter what activation pathway is taken, all three ways that the complement system acts to protect you will become activated. So when C3 splits into A and B, both A and B have functions. But first, let's follow A. C3A is a very potent stimulator of the inflammatory response. So C3A is going to activate mast cells to release histamine, which is the main organizer of the inflammatory response. C3A also increases vascular permeability, making your blood vessels even more leaky, which is going to attract more cells and more complement proteins to the area. C3b, on the other hand, embeds or sticks to bacterial cells. And it acts as what's called an opsonizing agent or opsonin. What an opsonin does is it stimulates phagocytosis. So you have some cells, dendritic cells, macrophages, that are sentinel cells and which can, if they recognize a bacterial cell or a virus through their toll-like receptors, can just phagocytize them. But most of your cells, that phagocytes, the neutrophils, they don't. They can only recognize bad guys when those bad guys have been labeled. And even macrophages and dendritic cells will be much more prone to phagocytize faster and more aggressively if the evil cell, if the bad cell has been labeled. And when we say labeled, what we mean is that it's been coated with something that is an opsonin. C3b is an opsonin. It's not the only opsonin your immune system uses, but it's a very potent one. So any cell that gets coated with C3b effectively has a giant eat me sign on it, and your immune system cells oblige quickly. So anything that gets labeled with C3b gets eaten pretty quickly and killed off. So that is the second way that the complement system acts to protect you. C3b sticks to bacterial cells and acts as an opsonin. C3b will also combine with C3 convertase to form C5 convertase, which again is going to split C5 into C5a and C5b. In the complement system, whenever you split something, you split it into an A half and a B half. Again, the A half, C5a, is a very potent inflammatory response stimulant. In addition to doing all of the things C3a does, it also acts as a potent chemoattractant or chemotactic agent. So it attracts white blood cells to the area, increasing their concentration so that they can attack all of the bad stuff that's around. Also stimulating the inflammatory response and increasing vascular permeability, just like C3a did. So both C5a and C3a act through the inflammatory response. C5b will embed in the membrane particularly of gram-negative cells. And it starts a cascade. So C5b embeds in the membrane, and it, what we say, recruits or attracts the rest of the complement proteins, C6, C7, C8, and then a whole passel of C9s. All of these proteins complex together in the membrane, forming something called the MAC, or membrane attack complex. I like to think of it as the Mac attack, because it rhymes. So the C5b will initiate the Mac attack, which forms this pore in the outer membrane of mostly gram negatives. And that pore pokes a big hole in the cell. Cells don't like it if you poke holes in them. They often die. Even if they don't die, that hole makes it so that like toxic elements like peroxidases and things like that can easily get into the cell and like kill the cell from the inside. So the Mac attack makes all of the other weapons that you use against bacteria more potent if it doesn't just kill the cell outright, which honestly it usually does. So this is lysis of foreign cells by the Mac. So that's the three ways that the complement system acts to protect you. C3a and C5a stimulate the inflammatory response. C3b is an opsonin, which increases phagocytosis, and C5b helps to form initiates the formation of the MAC, which will directly lyse foreign cells. And this is basically just what I just told you here. Yeah, so important things to keep in mind and remember: the complement system are proteins, not cells. You have 3 ways to activate it: the alternate pathway, lectin pathway, and the classical pathway. You have 3 ways that it helps to respond by inhibiting infection in foreign cells, through stimulating inflammation, through opsonization, and through direct lysis of foreign cells in the MAC. All right, thank you much, and that should be good.